All right. So what we're going to do for the next couple of days, starting today, we're going to now focus on the anatomy. So what we've been doing for the last 2 weeks, or the last 2 lectures, we've looked at how neurons work. So the first lecture, remember, we said we have neurons and glial cells, and we said, all right, well, how do neurons work? We talked about the gradient potentials and the action potentials. And now what we're doing is we're moving actually into the anatomy, the stuff that you're going, okay, this stuff I can grasp. Well, a lot of this is going to be, there's quite a bit of memorization here. Again, this would be ideal if you draw pictures to help you understand what's going on. And really, we're just going to be kind of walking through the different parts of the brain, pointing out the different things that they do. And so our starting point here is the entire central nervous system. The central nervous system, remember, is the brain and the spinal cord. All right? And so when you think of the brain, you really think of this big, large structure. This big, large structure kind of looks like a mushroom, I guess, on top of a stalk. And really, this is only just a small portion of it. All right? Technically, this is a telencephalon, which you don't need to know. I'm making it easy for you. But this is the cerebrum. All right? And so if you think of the brain, it really has these 4 different regions. It has a cerebrum, which is that larger structure. It has a cerebellum, which is the small structure that hangs off the back end. We have the brainstem, which is what is going to become the spinal cord. And then the area between the brainstem and the cerebrum, which after we slice in half, you can see that would be the diencephalon. And we're going to come back and we're going to look at these in more detail, but just to kind of give you a sense of where we're going, we're going to be kind of working from that larger structure of the cerebrum, and then we're going to work downwards towards the spinal cord. So, having said that, we can kind of say, all right, well, what does the brain look like? What does the cerebrum look like? Well, the cerebrum is divided into 2 halves, and so what we say is that there's these 2 hemispheres. There's a left hemisphere and the right hemisphere. Not really easy to see it from the view up top. Actually, that's a view of the cerebrum from the bottom side, so you can see the brainstem and the cerebellum. And then from the top, this is what it looks like, and you can literally see those 2 hemispheres fairly well. And then, of course, if we look at the cerebrum itself, and we look at each of the individual hemispheres, anatomists have broken down these hemispheres into separate lobes. And so what we say is that there are 5 lobes. Back when it depends on actually who you talk to. If you talk to anatomists, they say there's 5. Most people just kind of point and say there's 4, but we're going to go with the 5. And so these are really, really easy. All right? They're named, except for the 5th one, are named for the bones that cover them. So we have the frontal lobe, we have the parietal lobe, we have the occipital lobe, we have the temporal lobes, and so there's one on each half. And then if you take the temporal lobe and you separate it from the frontal lobe, that's where you're going to find the insula. It kind of lies inside and in between, which is why it's kind of like not a real obvious lobe. So the insula is the one that's kind of hidden away in between. We're going to come back and we'll see pictures of this a little bit better. So this is our organization. All right, 5 lobes. They represent where the bones that cover them: frontal, parietal, occipital, temporal, and then the one that doesn't make sense, which is the insula, which is hidden underneath. Structurally speaking, anatomists have looked at the brain and said, oh, look, there are landmarks. And so we're going to name the landmarks. The landmarks are, in real simple terms, are going to be these 3 basic structures: the gyrus, the sulcus, and the fissure. All right? The plural is gyri, the plural for sulcus is sulci. And really what the anatomists have done is, as they're trying to map the different parts of the brain, they use these structures to help them understand their location with regard to the cerebrum. All right, so a gyrus simply is a bulge. So every time you look at one of these things and you see the bump portion, that's a gyrus. And then between the bumps, between the bulges, are the sulci. So these are just kind of these depressions in between. And then they have these deeper folds, which are referred to as the fissures. Now, what distinguishes and how do you define which is which? That's a harder question. I think initially they were just saying these really, really deep ones are the fissures, and then some of the fissures they've gone back and renamed as sulci, so just whatever they point to and say. But in a real general sense, fissures and sulci are the ones that go deep. The ones that bulge upward are the gyruses, or the gyri, excuse me. Now, we haven't used a lot of anatomical terms, but there is one very specific for neuroanatomy, right? Or two terms that are specific for neuroanatomy. Rostral versus caudal. We've talked about ventral versus dorsal before. We don't, like I said, I tend to be very, use vernacular terminology here. But rostral and caudal are two terms that you'll hear neuroanatomists use. And really what they're referring to is locations similar to anterior, posterior, ventral versus dorsal. And it's towards the nose would be rostral, and caudal is towards the tail. And so you can think about your brain is like so. So the rostral portion is towards the nose, and then this is back towards the tail end of your body. So that would be caudal. So if you're doing slices or dissection, that's how you refer to where these structures relative to each other. That's a rostral structure, or that's a caudal structure. So be familiar with those. I'll probably just throw it on there and say, what is rostral? I'm not going to ask you what is rostral to this, but the idea is, can you use that terminology? So if you look at the brain, basically we divide it into two different areas. We have gray matter and white matter. Now, simply put, gray matter is where you're going to find the cell bodies of structures, or the cell bodies and the dendrites of cells. So when you're talking about neurons, really, that's where you're going to find their cell bodies. Whereas white matter is these areas where you're going to find axons traveling between points. And the reason white matter is white is because most of our axons typically are covered by what material? Glial cells, but specifically starts with an M, ends with Lin. Myelin, there you go, see. And so myelin is basically a cell that's wrapped itself in, myelin is basically cell structure that's wrapped around the axon, and basically it's fat. How many of you guys have ever been to a butcher? Well, probably not a butcher. How many have been to the grocery store and looked at meat? All right, there you go. That's the easy way to do it for the butchers nowadays. All right, when you go and look at a nice piece of ribeye, ribeye is very, very marbled. It has lots of fat in it. What do you see? You see that red meat, and it's marbled with what color? White. All right. Fat is white. Myelin is primarily fat, so white matter is white because it's primarily fat. It's basically those phospholipids of those other cells. And so that's why it kind of stands out. Now, gray matter doesn't look really gray. It's really more of a beige, but I guess they didn't have a term for that. So basically you have this beige area and this whiter beige area. All right, and so that's what these two areas represent. Gray matter represents the cell bodies, whereas the axons are going to be represented by this white matter. Now, if you look at the central nervous system, what you're going to see is that there's a high degree of organization as to where you're going to find cell bodies and where you're going to find axons. In other words, the gray matter and the white matter are in very specific locations. And so what they've done in this picture right here, you can see is that they're trying to show you different slices. All right? So the A, B, C, and D. So there's a slice through the cerebrum, there's a slice through the brainstem, there's a slice through the brainstem again, and then down here is a slice through the spinal cord. And if you look at this, you can see, for example, here is the spinal cord. And out here, you can see white matter. On the inside, that's where the gray matter is located. All right? So in the spinal cord, gray matter is found centrally, and then outside of that gray matter is the white matter. So you can imagine that here is where cell bodies are located, so that means we're going to have processing of information. White matter is where you're going to have axons, so that's where information is traveling. So this is where we're going to find tracks. In other words, the material or the information is going to be sent up and down or left and right within the spinal cord. As you move upward, so here we are in the medulla, and you can see there's that gray matter again. All right. This right here is a ventricle. There's a central canal right there. It's hard to see. And then you see all this white matter. And then embedded in the white matter now are areas of gray matter. So even at the level of the medulla, at the brainstem, we have gray matter that's kind of centrally located with white matter on the outside. And then as you move up to the cerebrum, you can see that there's gray matter. See the two ventricles, they're kind of paired in the center there. And then you see these splotches of gray matter everywhere, so that would be that internal gray matter, surrounded by white matter, which is the lighter stuff. But then, once you look at the cerebrum, on the outside, you have another group of gray matter. So it's kind of like an Oreo, gray matter, white matter, gray matter. And so what's happened now is that we needed more areas of processing. So in the cerebrum, we have lots and lots of processing take place. And so what's happened is that we've reorganized, so the gray matter is not just centrally located, we've added more cells on the outside, so there's more processing. This is referred to as the cortex. So when you think of the cerebral cortex, you're thinking of that outer layer of gray matter. Inside would be white matter, and then you have these other areas of gray matter. Those other areas of gray matter are called nuclei. So nuclei in the central nervous system refers to areas of cell bodies where processing is taking place, surrounded by gray matter, surrounded by white matter. And I'm ignoring, of course, the cerebellum, but the cerebellum is kind of the same thing. Gray matter central, white matter, gray matter on the outside. And that would have cortex as well. All right. So, when you think gray matter, what are you thinking? What do you think of? Cell bodies, processing information. When you think of white matter, what do you think of? Axons, tracks, whatever. And then you're thinking of information traveling. All right? How do you guys think your brains are important? Were you guys getting your kids with all the special announcements like, you know, your brain on drugs and stay away from stuff? Did you do that? We had the frying pan with the eggs in it. You guys are probably too young to do the frying pans with the eggs. All right. You want to protect your brain, right? How many guys are scared to death of concussions because of all the, there's scared to death of concussions. I mean, nowadays, when we were kids, when I was a kid, there was no such thing as a bicycle helmet. You wore bicycle helmets if you're riding motorcycles, and only if you were doing stunts. All right. Nowadays, you get on a skateboard, you have to wear your helmet, you have to put on your pads, you have your mommy write you a note to say it's okay. That's right, that's that. Some people are smiling at me. Would you say your brain's important? Would you want to protect it? Yeah. Good news, your body already protects your brain. It does a really good job of it too. There's actually multiple levels of protection. And what we say is that there's 4 levels of protection. Now, the obvious one is going to be the bone. All right? We have the cranium. The cranium, we've already talked about, it surrounds the soft tissue of the brain. And so that protects it there. And in terms of the spinal cord, you have the vertebrae. All right? And so we have this outer hard shell that serves to protect the squishy inside. I want to kind of diverge here for a second. How many of you guys have a sense of how soft the tissue of the brain is? Anyone have any idea? Anyone ever seen a brain? It's like jelly. That's actually a good way to put it. The best way I can describe it, it's like, how many of you guys have ever worked with margarine or butter? All right, that's what it's like. It literally just, it's soft and squishy. You know, if you guys watch Big Bang Theory, you'll see every now and then Amy working on a sheep's brain. She actually has her PhD in neuroscience. The actress does. She was Blossom. She was a teen actress. Then she went on, got her PhD in neuroscience, and then they hired her for the show just like on a whim, and so that's how they kept her on. But she actually has her PhD in neuroscience. And so whenever you see her working on a brain, that's stuff that she actually did. You know, kind of cool. I guess. I don't know, maybe geeky. All right, but anyway, when you see the pictures, when you see them working on the brain, they're kind of hard and stiff. And you got to think, well, why is that? Well, they fixed it in alcohol, basically formalin is what they fix this tissue in. So it's hard and stiff. That's not what brain is like. It literally just falls apart like warm butter. And so you need to have these structures to hold everything in place to make sure it doesn't just kind of ooze everywhere. All right. So that's kind of the outer portion, is that cranium, that bone that serves to protect. All right. Now, surrounding the actual tissue itself, this oozy tissue, is what are these 3 layers of just rapid. They're called the meninges. All right. Singular form is meninx, M-E-N-I-N-X. So if you see me say meninx, that's what it's referring to. So we're going to have these three protective membranes that protect. And then embedded between those membranes and surrounding the brain itself, we're going to have cerebral spinal fluid, which is basically a layer of fluid that surrounds and protects the brain. And then we're going to have a fourth layer. The fourth layer, which is referred to as the blood-brain barrier. Now, the blood-brain barrier is a little bit different. What we're doing here is not so much of a physical barrier, although it is a physical barrier, but it's not protected like the other ones with regard to trauma. But it's a physical barrier to prevent materials from moving from the blood into the cerebral or into the brain tissue. And so it's not only physical, but it's also a barrier that serves kind of like as a chemical barrier. It prevents foreign substances like bacteria, viruses, and other horrible things, but also just chemicals. Some very simple chemicals like water can be very damaging to the brain tissue because now what you're doing is you're altering ion concentrations and the cells won't properly behave. So we'll talk about all of these as we go through. But we're going to ignore the bones, just know that the bones are the outer layer and that they serve as that hard protective barrier. They're kind of your helmets. All right. Now, with regard to the meninges, the easy way to think about this is that there are three layers, and from the inside to the outside, based on their name, they create a pad to protect. I would say working outside to inside, but that makes absolutely no sense. So the inner layer is called the pia mater, the middle layer is called the arachnoid mater, and then the outer layer is the dura mater. All right, so we're going to be kind of working backward. We're going pia to arachnoid to dura, which is going inside to outside. All right. So with regard to the pia mater, all right, and we're creating this pad. The pia mater is very, very thin. It's highly vascularized. In other words, it is the last barrier that covers the surface of the brain. It is actually so thin and so fine that it can actually follow the gyruses and the sulci as you move up and down. So you can imagine with regard to this, it's really hard to see the color here. But if you look right there, there's that boundary. There's a little purple line. The purple line goes down with the sulcus, comes out, and just keeps following along. And it comes down and goes back out. So that would be the pia mater. It's closely adhered to the nervous tissue. And this is what allows the small blood vessels to stay very, very close to the nervous tissue and ultimately penetrate into the nervous tissue so that the blood can be delivered to it. All right? But this is that first barrier. All right? And we say it's highly vascularized, so you're going to have the small blood vessels that are located there. Now, the layer that lies above this is called the arachnoid mater. Why do you think they call it the arachnoid mater? Say again? It's web-like, all right? Arachnoid, spiders, right? Arachnids. This is where the brain spiders live. One person smiled at that. Anyone here have brain spiders? Well, let's find out. You see the cartoon. The arachnoid mater, also vascularized, actually it's vascularized in between. This is the arachnoid mater here. There's a pia mater. You see the space, and then right there is where the arachnoid mater begins. I'm actually going to see if my little laser pointer works. Last time I came in here. All right. Guess I take it close up. So right here, that's the arachnoid mater. Okay. This was the pia mater. And you can see we have this space in between. Inside the space, you see all these little tiny trabeculae, these little tiny fibers that are holding these two things together. This is why we make the arachnoid matter, because it had all these trabeculae. It looks like a spider web. All right? So this is why I say that's the area where the brain spiders live. All right? Now, the space right here is actually called the subarachnoid space. It lies below the arachnoid. So if you have the arachnoid, you have the subarachnoid space, then you have the pia mater. The subarachnoid space is going to be filled with cerebral spinal fluid. And then, as you can see, we travel up and then we see these penetrations right in here, that penetrate through the dura mater and into what are called the dural sinuses. Now, the dural sinuses we'll get to in just a moment, but in essence, this is going to be filled with blood that's going to be returning back to the heart. So blood that has come into the brain, that's provide nutrients and stuff, is now leaving and going back to the heart so it can get its oxygen back in the lungs and get nutrients as it travels through the body. In essence, what's happening here is we are penetrating into that so that the cerebrospinal fluid that we're producing can actually have a way to leave the brain and be recycled. Right? So this structure that's penetrated is called an arachnoid villi. All right? So, subarachnoid space, there's the arachnoid mater. Arachnoid villi, cerebrospinal fluid is found in the subarachnoid space. The little tiny webs are the trabeculae. And then finally, this is also, you can see in here, you can see there's the vein, there's an artery. These are the things that are going to be dividing, and the arteries are getting smaller and smaller so that they can penetrate through the pia mater. The outer layer is the dura mater, and there's actually two layers, and this is something that you can actually experience at home. Anyone here have like gallon freezer bags at home? Yeah. Okay, go pull one out and go play with it. Tug on it, pull on it, stretch it, and stuff like that. That's what the dura mater feels like. Very, very tough, very inelastic, doesn't have a lot of stretchability to it. In essence, it serves as this outer layer. There's two layers to this, all right? You can see the outer layer, which is going to be the periosteal layer, so next to the bone layer. And then you have over here the meningeal layer, and you can see how it separates and kind of goes down. But notice that it just kind of covers the brain. It doesn't follow all the sulci and the gyruses. Right here, this is that central fissure, and so it's forming what is called the falx cerebri, which we'll get to in just a moment. But in essence, otherwise it just kind of overlays the entire structure. All right. Now, where these two layers separate, right here, that's where you're going to get the formations of these sinuses. All right, and this structure now basically creates this large pooling area, this sinus, that allows for blood that's leaving the brain to ultimately pool before it goes down through a vein back to the heart. All right. And as I mentioned, with the arachnoid villi, once again, right there, that's how you're moving cerebrospinal fluid from the subarachnoid space back into the blood. Now, the dura kind of serve as this tougher structure to help maintain the shape of the brain, to maintain the hemispheres so that the brain doesn't move around all that much. And so what we end up with are these structures, these extensions that allow the brain to kind of sit. All right, so it partitions the brain and holds them in place. All right, and these are what are referred to as the septa. There are four cranial dural septa that you need to know. You can see their names up here: the falx cerebri, the tentorium cerebelli, the falx cerebelli, and the diaphragma celli. All right, now you're looking at that, you're going, those are big, scary words, and I don't understand what you mean. I don't want to know what they mean. All right, let's keep this simple. Cerebri, what do you think that has to refer to? Cerebrum. So there you go. There's the easy one. And the cerebelli? Cerebellum. All right. So you already know that the name already tells you a little bit about where it's probably located. You think the cerebelli is located or associated with the cerebellum? And the cerebri is located with... Okay. So far, we're halfway there. Now, you're sitting there looking at falx. I have no idea what falx is. I didn't know falx is easier, so whenever I don't know something, I just type it in the computer. Pops up. Falx means sickle. You guys know what a sickle is? All right, it's Halloween time. You should know what a sickle is because one of the main characters of Halloween is Death. Death is that big black robed figure that walks around with a sickle. Right? That big whole thing. And what's he doing? The reaper is reaping, using the sickle to cut down, well, in the case of death, life, but basically to mow down wheat. All right? And the shape of a sickle, wide at one end, and it gets small and thin at the other end. What do you think the falx cerebri and the falx cerebelli were named after? I mean, based on that. The shape? I'm going to stare at you until you nod your head one way or the other. Okay, there we go. Okay. Yes, all right. So you can see there, it has this kind of shape like a sickle. All right. So that's one. All right. What about tentorium? What do you think tentorium has to do with? If you had to guess, if I put a gun to your head and said, your life or death question here is, what do you think tentorium has to do with? What would you say? Here's the gun. Come on. Tentorium. Tent. Covering. Tents. Doesn't that look like a tent? Where do you think tent came from? Some guys are sitting around saying, let's find a 4-letter word that means camping. Tentorium. It's to cover. All right. And then the last one, diaphragmacelli. That's a little bit more difficult. Diaphragm is something that is a hole. Celli means seat. All right. Again, I don't have a lick of Latin in my background. Literally, never taken a Latin course or a Greek course or anything. If I don't know something, I look it up. And I'm like, well, how do I remember this? Well, the diaphragmacelli, we're going to look here in just a second, basically shows is a hole that allows the infundibulum of the anterior and posterior pituitary to pass on. All right. So I just want to point out the name's going to tell you what they do or what they look like. If you don't know, just kind of look it up and oh yeah. All right. So the falx cerebri, you can see it right here. It's a big structure. It goes right down the central fissure, and what you're doing is you're basically creating the division between the left and the right hemispheres. All right, so your left and your right hemisphere are defined by that falx cerebri. All right, now within the falx cerebri, we have 2 sinuses that are important. All right, we have the inferior and we have the superior sagittal sinus. Now, sagittal should make sense because we've learned the term sagittal. And so here we go. We've got one up here and one down there. Those are your 2 sinuses. Which one do you think is superior? The one on top, right? Because superior means awesome. No, it means above. All right, so we have the superior sagittal sinus. We have the inferior sagittal sinus. And those are the structure that are going to ultimately take blood away from the brain and join up before they leave. The tentorium cerebelli, you can see here. All right, so this, you can see right there, that's your cerebellum. That right there is the tentorium cerebelli. All right, so notice what it's doing. It's separating the cerebrum from the cerebellum. All right, that's its structure. Now, within the tentorium, you can see here that we have the transverse sinus. Sorry, it's very easy there. You can see right in there that would be the transverse sinus. All right. So what we're doing is we're bringing the sagittal sinuses together with the transverse sinus, and they're going to join together and allow blood to escape together. Then we have the falx cerebelli. Right there, they're showing you, and you can imagine it comes down and does that action. That would be the falx cerebelli. And then lastly is the diaphragmacelli, which is not an easy thing to see. Right in here. See that big giant gaping hole? All right. That should be the hole right there. That right there is where the pituitary gland sits down inside that bone. I'm just going to put it right now. Say it now. We're going to come back to it later. This is one of the most important glands in your body, and it sits about this big. All right. Literally, it's a little itsy bitsy tiny thing. It looks like a little tiny upside down button mushroom. All right. And you'll learn about this a little bit later because it basically pumps out every single solitary important hormone in your body. That's where it's located. And it actually hangs down and goes through that diaphragmacelli. Now, last little thing is the occipital sinus. You can see it would be coming down this direction, which you can see better in the cartoon. That's the occipital sinus in the falx cerebelli. So structurally, what we have here is we've got 4 different four different membranes that divide up the brain. All right, we have a couple sinuses that we need to know, and it shows how blood is leaving out through the brain. Questions about this so far? Do you think you could draw these? Do you think you can identify them? Yeah. Okay. So during development, I know I never talk about development, and this is not so important in terms of you need to know the development, but during development, the brain actually forms as a tube. All right, you create what is called the neural tube, and then it develops on and creates all these very interesting structures. And because it forms as a tube, what that means, it has a hollow center. All right, this hollow center is what ultimately becomes the central canal in the spinal cord and what becomes the ventricles of the brain. Now, the ventricles of the brain, their job is to produce CSF, cerebral spinal fluid. All right, and so we have these ventricles, and you can see in our little cartoons here, it's the kind of thing that look like these ram horns. It's centrally located, and within these structures, we have a special glial cell called the ependymal cell. The ependymal cell is what is responsible for producing the cerebral spinal fluid. All right, so they're closely associated with the blood. They line these structures. They take material, water, and other materials from the blood, and that's how they make the cerebral spinal fluid, and they put it into the ventricles. Now, there are 4 ventricles. The 4 ventricles are interconnected because this is one long tube that has been kind of malformed to create these structures. All right, and so the easy way to look at this is just kind of walk through them from top to bottom. So we have both a left and a right lateral ventricle. So that's the first one. So it's a paired structure. And again, you can see here kind of what it looks like from the front view. All right, but from the side you can see it's these longer structures. All right, and then between these, you can see how they connect right there. All right, they're going to come down and they're going to form this structure here. That is the third ventricle. So between the third ventricle and the lateral ventricles is the interventricular foramen. That's the connection between them. All right, so foramen means hole, between ventricles. So interventricular foramen down to the third ventricle. The third ventricle right here continues down to the fourth ventricle. That continues down is called the cerebral aqueduct. All right, so third ventricle communicates with the fourth ventricle via the cerebral aqueduct. Now, the fourth ventricle lies here between the brainstem and the cerebellum, and there's a couple of holes from which cerebral spinal fluid can leave and go into the subarachnoid space. These holes are called apertures. So we have lateral apertures. Lateral apertures would be on the lateral side, so there's one on the left and one on the right. And then we have a median aperture, sits right there, and that allows fluid to escape behind or below the cerebellum. And then that fourth ventricle continues downward as the central canal. It just keeps going down and down and down until it reaches the bottom of the spinal cord, and now you're opened up into the subarachnoid space. So what we have here is a structure in which the cerebral spinal fluid is going to be made. It goes from lateral ventricles to the third ventricle to the fourth ventricle, down, out, and around. And so we're dealing with the down, out, and around is the subarachnoid space. So this is kind of putting it off. Well, it's actually, you can't really see the ventricles here. This right there, that would be the third ventricle. This right here is a portion of the lateral ventricle. And there is the fourth ventricle. And what you're looking at here is that sagittal view where you can see the subarachnoid space. See? It goes all the way around, and it covers and protects the entire brain. It covers and protects the entire spinal cord. And so the cerebral spinal fluid is simply a clear, colorless fluid that's, it arises from the ependymal cells, fluid that has been taken from the blood plus other materials that have been taken from the blood and has been produced in the ventricles and ultimately filling and circulating around in that subarachnoid space. It's constantly being produced, and it's constantly being surrounding the brain. So it kind of serves as a cushion. And what we say is that there are three basic functions. It provides buoyancy to the brain, It provides protection, and it provides stability in terms of the environment for the brain. So let's kind of expand on that and see what we mean by it. All right? Now, with regard to buoyancy, the fluid has all these materials in it that give it density. And in fact, the density of this fluid is roughly the same as the brain. So when you take the brain and you put it into the cerebrospinal fluid, it sits wherever you put it. All right. Now, obviously, this surrounds the structure, so it's just going to be kind of equal all the way around the brain. But if you were to take a jug of cerebrospinal fluid, I guess a jug, I don't know, a container of cerebrospinal fluid, and you put the brain in it, if you put it on top, the brain would sit on top. If you took that brain and put it at the bottom, it would just sit there. If you took the brain and stuck it in the middle, it would just sit there. For that very reason, because it has the same density. It just holds and maintains the position of the brain. All right. Now, this is advantageous because, remember, the brain has mass to it. And so if you take something and it has the same sort of buoyancy, the same sort of density, that removes weight. All right. And so what we've done is we basically effectively reduced the weight of the brain by 95%. Now, this is beneficial. Because if the brain has mass, gravity acts on mass, and what happened is because I told you the nature of the brain, what kind of tissue it's like warm butter, right? And remember, if you can think about the cranium, we have this big giant foramen magnus back here in the back, and if it had weight, what would gravity do? It'd pull the brain down a hole, right? And so basically your brain would end up at the bottom of your back, and it would just would be there. So this helps support the brain. It keeps it in place because it no longer has weight. It serves as protection. All right. So you're familiar with your brakes. Most of your brakes, as you know, have liquid inside their structures. And so what happens, or your shock absorbers also have liquid inside them. And so what happens is when you have a liquid that is mostly water, it's non-compressible. And so what happens is that it can absorb energy very, very well. And that's what it does. It just serves as a shock absorber. So part of the reason you can run head-on and stuff and not get completely damaged is because that water absorbs a lot of that energy and disperses it outward and around. All right. In terms of environmental stability, this is a little bit more complex. And so there is this tight relationship between the cerebrospinal fluid and the extracellular fluid surrounding the nervous tissue. And what this means is that as the neurons and stuff produce waste, it can quickly shuttle that waste into that extracellular space, and then from the extracellular space, it can quickly move into the cerebrospinal fluid. Because the cerebrospinal fluid is always being moved away from the brain, then you can move waste very, very quickly. And as you can imagine, if you're trying to ensure stability, you want to have things normalized. And so by removing waste, you're maintaining that normal environment. So that's kind of what this is basically saying. It allows you to move materials very quickly to and from the tissue itself. The mechanisms that it's doing so, I don't want to get into. It's a little bit complex, and like I said, it's interesting, but it's just complex. The other thing I'd say is that it helps protect in terms of chemical fluctuation. So, for example, when your cells are very, very active, they actually get larger by a small margin. And so what happens is that actually has an effect on the surrounding environment. Basically, it reduces the amount of water that's available around the cells, which means you effectively increase osmolarity, which can be detrimental to how the cells function. And so the cerebrospinal fluid helps to adjust those chemical fluctuations. That would be an example of that. But like I said, it's a little complex. Now, how we go about making this stuff. All right. You can see in our little cartoon up here, there we go, right up there, that is inside, this is our third ventricle, there's our lateral ventricle, beginning with the lateral ventricle. And you can see up there, and down here in the fourth ventricle, the structure is called choroid plexus. Choroid plexus is a specialized area of the pia mater where you have the blood vessels penetrating very, very close to the surface of the ventricle. And overlaying the pia mater in these areas are ependymal cells. Ependymal cells surround the whole structure, but in this particular case, this is where those capillaries are located. And so what's happening is that you're bringing the blood right next to where it needs to be. So these are ependymal cells. That would be a capillary. This would be the inside of a ventricle. And so what's happening is these ependymal cells are pulling water and other nutrients, other solutes out of the blood and moving it out into the ventricle, forming the cerebral spinal fluid. All right? That's the structure. That's what we're doing. Now, with regard to what this fluid looks like, with cerebral spinal fluid, it's very, very similar to plasma. All right, so it looks like the fluid that you find in the blood. The difference is that we're being very, very selective in terms of what we're pulling out. So the ion concentrations are fluctuating. All right, there's very few proteins, just like you'd expect, but the ions and stuff, so it doesn't look exactly like plasma. It's similar to plasma, but there are ions and stuff that have varying concentrations. All right. So we're being made here in the laterals. We're being made here in the third ventricle. We're being made here in the fourth ventricle. So the flow of cerebral spinal fluid would be from here, the laterals, through the interventricular foramen, down into the third ventricle, down through this cerebral aqueduct, down into the fourth ventricle, and then out through the median or the lateral apertures, and then down through the central canal. And once we're out of those areas, either through the apertures or out through the central canal, that's when we're in the subarachnoid space. And we're flowing away from the central area towards the dural sinuses. And you can see there's a dural sinus. And what do we have at those dural sinuses are those arachnoid villi. So we're constantly producing the cerebral spinal fluid. It's constantly being pushed away from its site of production, and it's being pushed to where it can exit once again and join back to the blood. So the arachnoid villi serve kind of as the exit point. If the choroid plexus is the entry point to produce the cerebral spinal fluid, the arachnoid villi serve as the exit point for where the cerebral spinal fluid goes back to the blood. Does that make sense? You're looking at me like no. Well, let's kind of look through it again. All right. I wish I had, let's see. No one's up here. You make about 120, or the total volume of about 125 to 150 mils. Anyone here have a water bottle? You know, like a bottle of water? I don't. I know, my laser pointer sucks. I can barely see it here. Actually, look at your bottle. Your microphone? Oh, my microphone? You got it? Yeah. Darn, I guess I have to pop that down. Have another battery. Yeah, it died. How far back did it die? Oh, just when I talked. Okay, great. All right. So hold up your bottle real quick. All right. How much is this bottle? Anyone know? Okay, that's a good American answer. What is a good scientific answer? Half a liter, which is 500 mils. Your body produces that much cerebral spinal fluid on a daily basis. How much do you have with regard to the space? It's up there on the wall. 125 to 150 mils of cerebral spinal fluid at any given time is circulating around. That is 1/4 of that bottle. That's how much cerebral spinal fluid you have. You're literally replacing your cerebral spinal fluid on a daily basis 4 times. All right, does that make sense? Good. When I see one has this, that's wrong. All right, so you're constantly producing the cerebral spinal fluid. You're constantly making it as it moves from the lateral ventricles, into the third ventricle. It's being made in the third ventricle along with the lateral ventricles. And it's moving from the third ventricle down to the fourth ventricle. From the fourth ventricle, it's also being made. And then it's going out through those apertures, out into the subarachnoid space, down through the central canal, and out into the subarachnoid space. And it's exiting, because if I'm constantly making it, that means it's going to be making more and more and more. It's going to be expanding outward, right? And so I've got to get rid of it. Especially if I'm making enough to replace the amount of cerebral spinal fluid I have 4 times over on a daily basis. And so that's what the arachnoid villi serve as. It's the place where that cerebrospinal fluid returns back to the blood. Now, how do we get it to go in the right direction? Well, a couple of answers is actually pretty simple. The ependymal cells, they have cilia on their surface, and because you have cilia, they sit there and they beat. So they're pushing the cerebrospinal fluid in a particular direction. Secondly, because you sit up straight, just like we really told you, right? Sit up straight. Basically, your posture, basically the way that you stand or move, allows us to move the cerebrospinal fluid in a particular direction. So postural factors play a role. The third thing that plays a role, well, simply put, we're producing pressure. And so there's roughly a 10 millimeters of mercury pressure gradient that drives fluid from where it's being made to where it's exiting. It's not a lot of pressure, but it's just enough. So that's why it's going in that particular direction. Now, the blood-brain barrier is the last level of protection. All right? And the purpose of the blood-brain barrier, as I mentioned, it's a physical barrier, but it's also a physiological barrier. And its purpose is to ensure that material that could cause damage to the cells, in the nervous system, are not going to be allowed anywhere near those cells. All right? Basically, what it does is that we're going to allow for the specific exchange of materials through these cells so that we can selectively say this is something that we need or this is something that we don't want at all. So we're being selective as to what's going to pass through that barrier. And you can see here, I said the reason for this is because any selectivity in the brain interstitial fluid is going to have dramatic effects. This is where I pause and tell a little story that will make you very nervous. Okay? You guys remember when the Wii came out? Have I already told you this story? No? Okay, so the Wii came out, like 2008, 2006? Okay, 2006. And it was a sensation. No one expected how the Wii would sell. Because you had PS2s. PS2 was out at the same time, and everyone thought PS2 was going to be the dominant of the market, Xbox, whichever version at the time. They were fighting each other, and the Wii got released, and no one expected how popular the Wii was going to be. You could not find a Wii in the country to save your life. Now, most of you are sitting there going, I could care less about video games and whatnot, but it's important to the story because of the popularity of the Wii, there were contests and there were giveaways and all sorts of things going on. And there was this particular contest in Southern California that was taking place. And it was called I don't know what it was called, but I always think of it as the P-Fla-Wii contest. Don't tell my story. See, you already know the story. All right. But the rest of them don't. So, the purpose of this contest was drink a bunch of water, hold your urine as long as you can, and the person who holds it the longest gets to win the Wii. You've heard of these types of contests, right? It's kind of like the big pickup truck. Everyone has to touch the truck. Last person touching the truck wins. These things can go on for hours. And part of the strategy is figuring out ways to get the person to, mess it up. So you're like sitting next to the person who drinks a bunch of water. Think of waterfalls. Think of running water, showers, tinkle, 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 whatever it is, right? And so what happens is basically people who have to go to the bathroom are going to run and they don't get to win the Wii. Well, in this particular case, they had this small, petite woman who was trying to get the Wii, and I don't know what the volume of fluid that they had to drink, but she drank all this water very, very quickly along with everybody else, for the contest. And of course, when you drink water, your body's trying to find places to put it, all right? So it's going to equilibrate throughout the entire body, all right? So remember, when you drink, it goes into the digestive system. From the digestive system, it goes to the cardiovascular. From the cardiovascular, it gets distributed among all the tissues. Well, the blood-brain barrier is not particularly effective against water because water goes wherever there is a higher concentration of solute, all right? That's just typical osmolarity. And one of the places it went was to the brain. And because it went to the brain, it diluted out the solute she needed in order to get her heart to beat properly, in order for her to breathe properly. In essence, what happened was she had heart failure congestive heart failure, and she had respiratory failure because she drowned in the water in her own body. The blood-brain barrier tries to prevent things like that from happening, but obviously water is not one of the things that it can stop. The blood-brain barrier serves to protect you from things that can cause you harm, but not water. So, very, very valuable, very important structure. So, we say that it's an anatomical barrier. That would be a physical barrier. And we say it's a physiological barrier. Now you're all thinking about the Pee for the Week contest and like, oh, I better watch how I drink my water. So, what do we mean that it's anatomical? Well, we're talking at a very small level. So we're talking at the cellular level. And what's happening here is that as the capillaries, this is very early on in development, as the capillaries are going through, what happens is astrocytes come along and they connect with and attach to the outer surface of the capillaries. And what they do is they communicate with each other, and astrocytes tell the capillaries, hey, I need you to create tight junctions. And so the capillaries do. Now, throughout your entire body, capillaries have junctions between the cells of the epithelium, what we call endothelium of the capillaries. But the tight junctions throughout your body are fairly loose. Okay? In other words, they're leaky tight junctions, which is kind of an oxymoron, but that's just what we refer to them as. All right? And so when we say that they're leaky, that means you allow materials to pass in between those particular tight junctions. You can think about it like this. If you were to take water and put it in your hands and cup your fingers like so, would water leak out? Yeah, because you don't have tight points between all your fingers. There's some places where water can leak through. All right, so that would be an example of what's going on throughout the body in terms of capillaries. But in the brain, what happens is these astrocytes tell the capillaries, these endothelial cells, make these junctions tight so nothing can leak through. And by doing so, now what you've done is you force anything that wants to pass in through into the or leave the capillaries. It has to pass through the cells, the endothelium of the capillaries. In other words, you have to go through the cell. You can't go between the cells. And the only way that's going to happen is if you have the right receptors and the right carriers to move things across the cell. Now, on top of that, you have a very thick basement membrane. All right, remember, a basement membrane is simply the glycoproteins that are produced by the cells, the epithelium, and the connective tissue behind them. And that's how they connect to each other. So what you have now is you don't have this little tiny thin basement membrane. You have a thick basement membrane, which means if you pass through the cell, now you have to wind your way through all that material. And if you're too big, you're going to get stuck, and then you're never going to get through. So it limits what actually can pass through. And then on the other side of that basement membrane are the astrocytes, which are also closely associated with what you're seeing in these pictures. And so you can kind of see here what you have to pass through if you are a molecule leaving the blood trying to get in the tissue. You have to pass through the endothelium of the capillary, you have to pass through the basement membrane, then you have to pass through the astrocyte. And in order for that to happen, you have to have the right carrier, the right molecule to grab you, move you across each of those cells, and eventually allow you to pass into the nervous tissue. So it's an anatomical barrier. It literally is a gauntlet of cell to cell to cell in order to pass through. So if there's nothing to allow you to cross, you're not getting in. Now the reason water can pass through is that water can pass between cells and through cells without a carrier. And that's why I like using the weak contest. All right, how many of you guys are planning on pharmacology? In other words, you're planning pharmacy school. One. You're all liars. I know there's more than one person in here planning on pharmacy. All right, so here's a question since you're brave enough to raise your hand. All right. If you're prescribing, or not prescribing, but if you have a drug that is going to work on the brain, what type of drug do you think it's going to be? Do you think it's going to be water soluble or lipid soluble? Lipid soluble. Why? If it's water soluble, is it ever going to pass through the cell membrane? No. But if it's lipid soluble, what's it going to do? La 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 la, I can go wherever I want. Yeah. So basically anything that's acting on the brain is going to be lipid soluble. So that's the anatomical restrictions. And you can see the answers up there too. Okay. Answers aren't always on the board. All right. With regard to physiological restriction, now we're just basically saying, are you water soluble or are you lipid soluble? If you're water soluble, you have to be transported. You have to be carried from one cell to the next, and you have to be moved across that barrier. All right. Each side of the plasma membrane. So passing from the blood, you have to pass the plasma membrane to go inside the endothelial cell, and you have to pass the plasma membrane to go to the basement membrane. Pass through the basement membrane, you have to pass through a plasma membrane to get inside the astrocyte. To get out of the astrocyte and go into the extracellular space of the brain, you have to pass through the plasma membrane again. So every time you pass one of those, you need to have something to transport you across if you're water soluble. If you're lipid soluble, you can do that yourself. That's not a problem. But of course, you have to survive in the water environment. And as I mentioned, water can diffuse happily. So the blood-brain barrier serves as this protective layer to ensure that the specific nutrients that you want or need can pass through, and the stuff that you don't want can't. Now, the way this happens, the astrocytes, as I said, they're the ones doing the work. They're the ones talking to the capillaries. If it weren't for the astrocytes coming along and saying, all right, capillaries, I need you to create these tight junctions, then you'd be able to pass in between the cells. And they also play a role because you have to pass through the astrocytes to get the nervous tissue as well. However, you do not have blood-brain barrier throughout the entire brain. There are specific areas in the brain where there is no blood-brain barrier. And this is to your benefit. First off, the hypothalamus is a regulator of many different systems in your body. And it needs to know, for example, what it needs to control. All right? In other words, if I don't know what's in the blood, how do I know how to control the things that are going on in the rest of the body? So it needs to be able to sample the blood to figure out what it needs to do. And if you had a blood-brain barrier in those particular locations, it wouldn't be able to do that. It's not the entire hypothalamus, there's just particular areas. The pineal gland also outputs hormone directly into the blood, so it has to have an opportunity to do so, so you're not going to have a blood-brain barrier there. The most likely way to poison your body is basically by putting something into your digestive tract. Would you all agree with me on that? Do you guys put things in your mouth right now? Maybe you guys say, no, I haven't. Are you sure? Have you ever stuck a pin in your mouth? Have you ever played the 5-second rule? Here's my cookie. It's okay. Right? Have you ever just tried something? Well, I'll give it a whirl. Right? Most common way to get something bad in your body is by consuming it. All right? That's the most likely way that you're going to get foreign invasion. All right? And so when your body starts detecting toxins in the body, because the way that you're going to detect them, they'll be circulating in the blood, is the body, the first thing it's going to say is, aha, I know where this came from. You consumed something you shouldn't have. And so what I'm going to do is I'm going to just throw up whatever in the stomach, and that will help me get rid of whatever the toxin is. So the vomiting centers of the brain are constantly monitoring the blood to make sure you don't have horrible, nasty toxins circulating through. And then the choroid plexus also needs to be permeable. And here we have those ependymal cells. They're not the blood-brain barrier, but what they're doing is they're acting as the mediators as to what can come allowed or be allowed in. Are there any questions so far? Is this scary? How we protect the brain? What are the 4 ways to protect the brain? All right, and then the membranes, and then the cerebrospinal fluid, and then the blood-brain barrier. Pretty easy, isn't it? So all you got to do is protect. When we come back, Yes, yeah, there. That's when I get the applause. Thanks. When we come back, what we're going to do is we're going to start looking at the structures inside and start seeing what they do. When is your exam? Next Thursday. So if you haven't signed up for your clock, just go ahead and grab your clock. Sorry.